Hello everyone, welcome to 10-Minute Physiology. In today's video, I'm going to give you a short explanation as to how hydrophilic solutes are exchanged at the capillaries. So with that, let's give it a go. So in order to understand how solutes are going to be exchanged at the capillary, we have to understand a concept called flux. So in order to understand flux, what we have here is two compartments, a compartment of blood and a compartment of interstitial fluid that is separated by a semi-permeable square membrane. Now this square membrane has a certain area that we can calculate by multiplying the length and the width. So the area is going to be x squared. So let's just say we have a certain molecule called A. So A has a certain concentration in the blood and a certain concentration in the ISF. And let's just say that the concentration of A is greater in the blood than it is in the ISF. So as a result, the molecule A is going to move from the blood into the ISF. Now from this, we can calculate the flux. So the flux, simply put, is the amount of molecule that passes through a certain area per unit time. So we can use or calculate the flux of a particular molecule by using Fick's law of diffusion. So this right here is Fick's law of diffusion, and in this equation, J represents the flux, P represents the permeability coefficient, and this represents the concentration gradient. Now from this equation, we can see two main things. The first thing is that in order for there to be net movement of a molecule, there needs to be a difference in concentration. If you don't have a difference in concentration, then there's no net movement. The second thing that we see here is that the flux is going to be directly proportional to the concentration gradient. So the greater the difference in concentration, the greater the flux. And P is the permeability coefficient, and it basically it's going to be different for every solute, and it basically describes how well the solute can pass through that semi-permeable membrane. Now, since we understand what flux is, let's talk about something called an extraction ratio. So what is an extraction ratio? Well, in order to understand that, we have a figure here. And in this figure, we have an arteriole and a venule, and these two vessels are connected by a capillary. And let's just say in the arteriole, we have 10 molecules of X. So when these 10 molecules of X enter the capillary in this first segment, we can imagine that 2 molecules of X are going to be lost to the ISF. So 2 molecules of X diffuse from the capillary into the interstitial fluid. Now let's just say the remaining molecules of X, so now we only have 8, move to the next segment. And in the next segment, we lose another 2 molecules. This will leave 6 behind. And then when the remaining 6 go to the last segment, we lose another 2, and this would keep 4. And then the 4 will move on into the venule. So from this, we can actually measure something called the extraction ratio. And what the extraction ratio does is it basically tells you how much solute diffused from the capillary to the interstitial fluid. So using this example here, we can calculate the extraction ratio by taking the original concentration of X, so remember we had 10 molecules at first, and subtracting from that original concentration the remaining concentration, so the concentration of X that we have in the venule. So remember the original was 10 and the final concentration was 4. And then we divide that difference by the original concentration, in this case 10. And when you do that, you get an extraction ratio of 0.6. So in other words, 60% of the solute that entered into the capillary was lost into the ISF. So 60% of the molecules that entered into the capillary diffused into the interstitial fluid. So this right here is the general equation for the extraction ratio. So it's the difference between the concentration of X at the arteriole and the venule, and divide that by the concentration of X at the arteriole. And this particular equation only looks at a single capillary. So it only looks at a single capillary. So how can we take this concept of the extraction ratio and apply it to whole organs? Because when we're talking about physiology, we're not usually talking about just one capillary, we're talking about a whole organ. So in order to do that, what we use is this equation. So this complicated equation has many different variables, and we're going to go through each of them. So the first one here is E, that is the extraction ratio. This is just the number E. And then here in the exponent, you have the permeability coefficient multiplied by the surface area over the flow. So what does this equation actually tell us? So the first thing that we're going to look at is the flow. So when the flow is really fast, or in other words, when the flow rate is at, let's just say it's infinite, so it's really, really fast, what's going to happen is that the exponent is going to go to 0, because whenever you divide something by a really big number, that number goes to 0. So therefore, anything to the 0th power is going to be 1. So 1 minus 1 is 0, so therefore the extraction rate is 0. So what does that mean? That means that when the flow rate is really fast, the extraction ratio is going to decrease. And that's because when a fluid is moving too fast, it doesn't give the molecules a chance to diffuse into the neighboring compartment. So in other words, the faster the fluid flows, the lower the extraction ratio is. The second factor that you see with the flow rate is that if the flow is very slow, then what happens is the exponent goes to infinity. And in this case, the e to the negative infinity power, so e to negative infinity, is going to be 0. So 1 minus 0 is 1. So in other words, 100% of the solute would go into the ISF. So in other words, what this is saying is that as the flow rate decreases, the extraction ratio increases. Because when the fluid flow rate decreases, what happens is it gives a chance for the molecules to diffuse from the capillary into the interstitial fluid. The next thing that we see here is the permeability coefficient, and we see that it's at the top of the ratio here. So the greater the permeability coefficient, the greater the extraction ratio, because remember, the permeability coefficient tells you how easily the molecule passes through a membrane. And then the last one is the surface area. So the surface area is actually correlated to the number of open capillaries. Now, in a particular organ, most of the time you're not going to have all the capillaries open at once. So usually when an organ becomes more active, more capillaries open. And as you increase the number of capillaries that are open, you increase the surface area, and therefore you allow more solute to be released into the interstitial fluid. So those are the main things that you should get from this equation. So the last thing that I want to talk about is how these molecules are actually transported. So in order to understand that, let's look at this figure here. So these two red lines are endothelial cells that are adjacent to each other. And what we see here are a couple of different things. So this thing that looks like a ladder is known as a tight junction, and these tight junctions basically lock cells together, and they can form something called gap pores. And these gap pores are really small pores that allow certain sized molecules to pass through them. And then these little blue rods in front of the tight junction, facing the lumen and facing the ISF, are going to be known as glycoproteins. And these glycoproteins form the glycocalyx. So these sort of act as a pre-filter that prevents certain molecules from passing. So in order to understand how the glycocalyx works and how the gap pore works, let's bring in a molecule. So this right here is a protein, and as we can see here, the protein is fairly big. So when the protein gets near the glycocalyx, you can see that the glycocalyx prevents the protein from going through between the cells. So the protein will reflect right off of it. Next, let's look at glucose, which is a lot smaller than the protein. And what we can see here is that the glucose passes right through the glycocalyx, through the gap pore, and as well through the other glycoproteins that we see at the other end. And water can also pass through easily as well. So in other words, the glycocalyx and the gap pores, which are made by tight junctions, allow small molecules to pass. but larger molecules like proteins are usually kept inside. Now, it is possible for some larger molecules to pass through the capillary, but they go through a special process called transcytosis. This is basically when you have vesicular transport. So you'll have these pits that face the lumen of the vessel, and what happens is these big molecules go into these pits, and it forms a vesicle, and then the vesicle travels to the opposite side of the cell and releases the contents into the interstitial fluid. So in summary, we talked about how small hydrophilic molecules can exit the capillary via paracellular mechanisms. We talked about how glycocalyx and the gap pores basically filter specific molecules, so they allow smaller ones to pass, but they keep larger ones inside. We also looked at Fick's law of diffusion, where we saw that the flux is going to be directly proportional to the concentration gradient and the permeability of the solute. And we also looked at the extraction ratio. We saw that the extraction ratio is proportional to the permeability, the number of capillaries that are opened, and we also saw that the extraction ratio decreases as the flow increases. So I hope this video helped you understand how different solutes are transported from the capillary blood into the interstitial fluid, and I hope to see you next time.